Okay, it's preparing for YouTube.
Yeah, so I think Shravanti is here. Sanjana, Shravanti is here.
Okay.
Can you please
Shravanti,
make
I'm letting Shravanti in. Shravanti, you start the streaming.
Yeah, and
I
make a
started
co-host
the
as well.
streaming, so I'll check on the YouTube also if it's.
Sorry, you start the recording. You start the recording, I meant. And please make Dr. Shravanti co-host.
Yeah.
Hi, Dr. Shravanti, can you hear me?
Can you hear me?
Yeah, I can hear you perfectly fine. Yeah.
Okay. Okay.
How
Hi.
are you doing?
I'm good. How are you?
I'm good, thanks. Long time.
I know, very long time. So where are you now, Sona?
I'm in US now. I'm in NIH.
Okay, in NIH. Okay. Okay.
Yeah.
Good to see you.
Good to see you too. Yeah. I don't see your video, I think, here, but I think you should be able to. Yeah, I can see it now. Yeah, perfect.
Video.
Yeah.
Right.
I just want to check if I can share my slides without problem.
Right. I think
should
we can do that. So
Yeah,
I mean,
should
while
I-
Sanjana, you are letting other people to join, we can check this
Yeah.
thing.
Or did Shravanti
Let's
want
go
to
through the
check
introduction.
first?
Sorry, I couldn't hear you.
Yeah. So I mean, we can introduce, do the introduction, and then you can share as well during that time.
No, I mean,
Okay.
if she wants to check, it's I think that would be better before we start letting people in.
Yeah. I think let's
Okay,
test
okay.
it. Yeah.
Yeah, sure.
Okay.
You can use the share screen option at the bottom.
Yeah. Do you see?
Yes, I can see it perfectly fine.
Yeah.
Okay. I'll stop sharing now and
Okay.
then
Yeah.
leave it there. Yeah.
And Devani can have the video up.
Devan, please reshare the video.
So I think we can, Sanjana, when you're ready, start allowing the people and we'll
Start
start.
letting people in.
Yeah, yes.
Is the
Okay,
video
thank you.
visible?
Yes, we are all good.
All good. So in 2 minutes, we will
Yeah.
start the introduction and all.
Just let's let people in. Yeah.
Okay, I let people in. So everyone mute and
Yeah. Sure. So I think in 1 or 2 minutes, we'll start the session. Okay, so hello everyone. Good morning, good afternoon, and good evening from wherever you're listening from. Welcome to the Biologically Speaking and the second day of HerStory of Science conference, where we are celebrating the exemplary achievements of women researchers in India. But before we start to the session, and while others are still joining, let me take this opportunity to express my deep gratitude to a team of enthusiastic doctoral and postdoctoral fellows from India, US, and Europe, who came together to design this academic interest group, Biologically Speaking, where we discuss the progress and prospects of biological research through regular seminars and blog posts. We are pleased to receive a funding from BiosWatch India that helped us to organize this 2-day conference during the weekend before the International Women's Day tomorrow. It is an opportunity to celebrate women's achievements, and today we are in the second and final day of this conference, where we bring to you 4 sessions spanning from health and disease to neuroscience and to biodiversity and conservation. We also have a special session this afternoon on gender equity in Indian academia, and we are joined by eminent speakers working on gender equity studies, along with science communicators actively working to reduce the bias, raise awareness against the bias, and take action for equality. We are truly overwhelmed by the response we have received from all around the globe and from different time zones, and we are grateful to you for your support, cooperation, and your active participation. We are also live tweeting each session during the conference from our Twitter handle at @BioSpeakWeb. Please follow us on Twitter and use hashtag HerStory to tag us and let us know how you're enjoying the conference. We are also proud to dedicate each session of this conference to a distinguished woman scientist involved with Indian science or influencing Indian scientific philosophy. So before we move on to the first session, here is a short video commemorating the women researchers, celebrating their achievements, and whom we are proud to host in this conference. So here it is, what we have experienced yesterday and what we look forward throughout the day. With that, welcome you all to the second day of HerStory conference, and I hope you all will have an enriching session today. And we move on to start our first session on health and disease, dedicated to India's one of the first female physicians of India, Dr. Kadambini Ganguly. Dr. Kadambini Ganguly, born in 1861 in Bhagalpur, Bihar, was the daughter of social reformer who campaigned for women's right to education. In 1878, she qualified the University of Calcutta entrance examination, and in the process became the first ever woman to do so. As she graduated from Bethune College, Kolkata, Kadambini, along with Chandramukhi Basu, became the first female graduates of India and the entire British Empire. Despite strong opposition from the then prevalent women oppressive society, she joined Calcutta Medical College in 1883. In 1888, she received her degree, becoming the first ever female physician with a degree in Western medicine in the whole of South Asia, and continued to increase her expertise in medicine, earning more degrees from Edinburgh, Glasgow, and Dublin. Being a married woman and a mother of 8 children, she had to dedicate significant time of her household to household work. She continued to manage her familial duties and pursue her academic interest simultaneously in an excellent manner, while also fighting against the oppressive society and being a beacon for women liberation. in honor of one of the first female physicians of India. Biologically speaking, is proud to dedicate this session on health and disease to Kadambini Ganguly, and our speakers include Dr. Shravanti Rampalli Deshpande from CSIR IGIB New Delhi, Dr. Karishma Kaushik from Savitri Bai Phule Pune University, Dr. Sheetal Gandotra from CSIR IGIB New Delhi. So our first speaker of this session is Dr. Shravanti Rampalli Deshpande. Dr. Rampalli Deshpande received her PhD from Savitribai Phule Pune University in 2005 under the guidance of Professor Shomit Chattopadhyay and a postdoctoral training from University of Ottawa. Thereafter, she joined InSTEM NCBS Bangalore as a Wellcome Trust DBT India Alliance Intermediate Fellow, and very recently she moved to CSIR, Institute of Genomics and Integrative Biology, New Delhi. Her lab's research interest is understanding the self-determination and the epigenetic regulation of somatic and stem cell plasticity and the role of methyl-dimethyl transferases in cellular differentiation. And I have a very personal note regarding Shravanti to share because I was privileged to work in a lab at InSTEM and CBS during my student days and get my first-hand experience to work on stem cell biology and for motivating me to take up this area of research. With that, it's a great honor to have Dr. Shravanti Rampalli as a speaker in this session. So great to see you after so many years and very much looking forward to your talk. Over to you, ma'am.
Thank you, Shaunak. It's indeed a great pleasure to be here. And I really like to thank the enthusiastic organizers who have put together this wonderful session. And now that I heard about Dr. Kadambini's history, I really feel privileged to be here. Being a mother of two, this is a fantastic place to be and to give a talk in this particular session. Okay, I'll share my screen and please someone let me know if you can see this.
Sure, yeah.
Can you?
Yes, it's perfect. Yeah.
Okay, okay. Once again, thank you for really great privilege to be here. So I'll be talking to you, just a minute. Why can't I move my slides? Yeah. So I'll be talking to you about this group of enzymes called as lysine methyltransferases. I have been interested in these enzymes for past 2 decades now, from my postdoctoral days, and now I still continue to work on these enzymes. Really what fascinates me about these enzymes is their absolute requirement in human development and disease, organismal development and disease. As you can see, there are many of them which are listed here, and you can see that they are a lot under investigation for clinical, preclinical studies also. Now, this tells you how important these enzymes are in the context of organismal development. Being a stem cell biologist, I have been intrigued about role of these enzymes in the context of development. And I have worked on several of these regulators since I was a postdoc and continue to work on those, as I said. But for today's talk, we will be focusing on this set of epigenetic regulators or lysine methyltransferases, which we call as euchromatic histone methyltransferases. So just to give you a brief information about what are euchromatic histone methyltransferases, these are present in dimers. There are 2 proteins, EHMT1 and EHMT2. They're very similar proteins. Both of them are enzymes, but EHMT2 is much more active in terms of enzymatic activity in the cell. These methyltransferases are recruited to the chromatin template by transcription factors, usually targeting factors. When they are recruited to the chromatin, what they essentially do is they methylate the histones, either mono or dimethylation. Now, when the histones are methylated at K9th position by these enzymes, what happens is the chromatin undergoes condensation. There are other proteins, helper proteins, which help condense the chromatin, join these group of enzymes. They are called as heterochromatic proteins, HP1s. When the chromatin condenses like this, it inhibits the transcription. Now, not only that these enzymes or their enzymatic activity inhibits transcription, it's been known in the field that when there is a canine methylation mark deposited onto the chromatin template, this chromatin eventually moves to the periphery of the nucleus. Now, what would happen if such a thing happens? This is what a cell would look like, a nucleus would look like. When canine methyl marks are deposited onto the chromatin template, it moves to the nuclear periphery, and what it does is it creates these domains, peripheral chromatin versus the central chromatin. The peripheral chromatin is usually condensed and silent one in terms of transcription, while the one which is present in the center is mostly very dynamic, active, and repressed based on the requirement of the gene activity at that time. So euchromatic histone methyltransferases are required for both dynamic activity and making the peripheral heterochromatin also. So we had been interested in this group of enzymes, as I said, for the development purpose. So what would happen if you don't have euchromatic histone methyltransferases? So we can't afford loss of these enzymes or complete loss of these enzymes. It causes embryonic lethality. But if there is a haploinsufficiency of these enzymes, people have noticed there is a neurodevelopmental syndrome called as Kleefstra syndrome. In addition to intellectual disability and other problems which are similar to autism, these people are obese in nature, they are male sterile, and etc. There are several problems which occur when there is a haploinsufficiency in terms of these enzymes, specifically euchromatic histone methyltransferase 1. So in my laboratory, when I started a decade ago, almost a decade ago now, we were interested to understand the spectrum of the proteins which these euchromatic methyltransferases interact with, and what is the mechanistic role in determination of cell fate by these enzymes. So we had performed a mass spectrometry analysis for identifying the novel partners. And today I'm going to talk to you about one such interaction which we have identified with a nuclear lamina protein called as lamin B1. I'll just give you a brief introduction of that protein too. Nuclear lamina, what is a nuclear lamina? Beneath the nuclear membrane, the organelle, which we all know, beneath the nuclear membrane, there is a matrix or lamina, which we call it as a nuclear lamina. Now, what is it made up of? It is made up of intermediate filament proteins, which are lamin A and lamin B, A type of lamins and basically B type of lamins. You all must know lamin A because of the progeria syndrome. Mutations in lamin A usually cause the progeria syndrome. Lamin B mutations are not known because generally it is believed that mutations in lamin B are not tolerated, so you don't tend to see them. Both lamin A and lamin B are critical for making the meshwork of the nuclear lamina, which is present underneath the membrane, and it gives the rigidity to the nucleus and help anchor the heterochromatin as well towards the periphery. It has also been noticed that during the context of aging, nuclear lamina proteins are less abundant or down-regulated during the course of aging. With this information, I'll tell you that what we have noticed or what is the mechanistic details which we could undertake for interaction of euchromatic histone methyltransferases with this nuclear lamina protein. The first thing which we did in our laboratory was to confirm whether EHMT B and lamin B interactions were real. So we performed a sequential immunoprecipitation experiment, and you would see that both EHMT1 and EHMT2 are present in complex with lamin B1. We have confirmed these results by in vitro interaction studies also, wherein we purified GST lamin B1. This is a recombinant lamin B1. When incubated with a set domain of EHMT, we see that EHMT set domain interacts with lamin B. We have done several other studies to prove this also in terms of expressing them in the mammalian GFP-based vectors. We also see the same thing, that EHMT set domain interacts with the lamin B1. Next, the obvious question was, what would happen if we were to knock down this enzyme? Is there any role of EHMT1 in maintaining the nuclear lamina integrity? So towards this, what we have done is we have transfected, these are human dermal fibroblasts, these are primary cells. We have transfected primary dermal fibroblasts with the control shRNA, and you would see that EHMT1 is present in the nucleus here, and the red staining is lamin B1. You can see that lamin B1 is present beautifully at the periphery of the nucleus, and some of it is also present in the nucleoplasm. It's known in the literature that some of the lamins are actually present in the nucleoplasm as well. So when we knock down this protein of our interest, EHMT1, what the striking phenotype which we found was the nucleus of knockdown cells was really small. Not only the nucleus size was small, but the shape of the nucleus was severely distorted as well, which indicated that knockdown of EHMT1 actually altered the composition of somewhere at the membrane level or at the lamina level. And we were hoping that it was at the lamina level because the interactions were with lamin B. What we have also shown, and which I'm not showing you today, is the levels of the nuclear lamin B when it goes down, which really doesn't alter lamin A to that profound effect what we see. So the next thing, I told you H3K9 methylation mark, which is this euchromatic histone methyltransferases deposit, actually targets the proteins targets the chromatin to the nuclear periphery. What I'm showing you is a TEM image where fibroblast cells were transduced with a control shRNA. What you can see is this is a nucleus here. The gray areas where you can see the cursor going down, this is the heterochromatin. And I'm hoping that you can see this, a black thread which is going around. This is a nuclear membrane. So beneath the nuclear membrane, there is a beautiful peripheral heterochromatin which is attached in these fibroblast cells. Now, our idea was to look what would happen if we were to knock down EHMTs, because our data has shown both EHMTs and EHMT1 and 2 knockdown actually results in change in the nuclear architecture, levels are altered, and the nuclear size is changed as well. So when we looked into knockdown of EHMT1, what we saw was something which was very interesting. What I'm showing you here in the cursor is a nuclear membrane. I hope you can appreciate that the nuclear membrane in the control cell is really a single thread beautifully moving across. But in SHEHMT1 cell, this became very crummy, as if someone has literally distorted that membrane. Other noticeable thing was the peripheral heterochromatin, which is present just beneath this membrane here in the control cell, is almost absent in these knockdown cells. Rather, we saw the floating islands of the heterochromatin in these cells. So we believe that the heterochromatin which is present in these cells is zipped off as a result of the knockdown of EHMT1. And that could be because of the effects of the changes in the nuclear lamina which we have seen. The next aim was to see whether EHMT2 or similar protein would have the same effect. When we did this experiment, what we noticed is, unlike EHMT1 knockdown, we really don't see a crummy nuclear membrane here. Nuclear membrane seems to be pretty much okay. There is zipping off of the heterochromatin. Substantial amount of it is moved inside, but there is still a lot which is there at the membrane. So what I'm trying to tell you here is there are 2 proteins which are highly similar, which are present in, which is thought to be present in the similar complex, but here are doing something which are 2 different things. While both of them function at the level of altering the nuclear lamina. So we believe that there are certain effects which EHMT1 functions independently compared to EHMT2, and that I can discuss with some of you independently if you are interested in. But overall, what we see is EHMTs interact with lamin B1, knockdown of EHMTs alter the levels of the lamin B1, alters the nuclear shape and the integrity of the nucleus. When we look in terms of heterochromatin formation or heterochromatin tethering to the periphery, we see that knockdown of EHMTs influence the tethering of the heterochromatin, especially more so pronounced in knockdown of EHMT1 compared to EHMT2. So the next experiment which we thought was, where would you see such changes in levels of the lamin B1? Is it real that loss of lamin B1 might be correlated to loss of EHMT in the natural conditions where we are not really messing around with any protein inside the cell externally? So towards this, we went to the aging model. We have gathered primary human dermal fibroblasts from different ages, which are indicated here. These are all ethnicity and gender control. All are female, quite Caucasian fibroblasts, primary dermal fibroblasts, which we have collected. And what you can see here is lamin B levels naturally declines from a very young state to adult state where everything is fine. But as one progresses towards aging, the levels further decline. Now, to test whether this is corroborated with the loss of EHMTs, or EHMTs, do they have any role in the context of loss of lamin B1, we looked at profiles of EHMTs also. And what you can see here, again, in the same set of the fibroblasts, is levels of EHMT2 here drops way before EHMT1, but complete loss of, or rather, loss of lamin B1 really at a larger level is correlated with the loss of EHMT here. So what this tells us is euchromatic histone methyltransferases exhibit a similar profile as lamin B1, but a substantial loss of lamin B1 really correlates when you have a loss of EHMT1. Now, if that is true, then what happens to the heterochromatin state in these cells? So again, we performed the TEM experiment. Here is a nuclei from 18 years old dermal fibroblast. You can see again a beautiful nuclear membrane, a thread-like a structure which is going around here, beneath which here is a peripheral heterochromatin, seems all well. At 37 years old, again, the nuclear membrane looks pretty much intact, and you can see that heterochromatin, substantial heterochromatin is present in these cells, while there is some amount of the loss of lamin B1. But when you look at loss of both EHMT1 and EHMT2 at, apologies, this is a 65 years old nucleus, wrongly labeled. You can see that membrane is fine, but there is no peripheral heterochromatin whatsoever. Almost everything is lost from here. Now, is this just a correlation? Or can we take this aged cell and put back to the heterochromatin onto its periphery if I were to overexpress euchromatic histone methyltransferases? Now, towards that, we first wanted to understand what is the mechanism by which EHMTs might be regulating lamin B1. Obviously, first one to look at is whether it is transcriptionally regulating, because EHMT1 regulates transcription. We saw that 50% of lamin B1 is transcriptionally regulated, but there was a substantial amount of the transcript still present. The next option for us was to look at, here is an enzyme and here is a substrate. Does EHMT methylate lamin B1? Towards that, we purified recombinant lamin B1. We incubated it with SET and SAM, which is its donor, methyl donor. And when we performed this reaction, what we realized is that euchromatic histone methyltransferase 1 and 2, which I'm not showing you today, both have ability to methylate lamin B1. So this is an in vitro reaction. Does this happen in vivo? For this, what we did is we took nuclear lysates, we immunoprecipitated lamin B1, and that immunoprecipitated material we probed for methyl probed with a pan-methyl antibody, lysine-specific antibody. And what I can show you is methylated, immunoprecipitated lamin B1 actually does show methylation of methylation signal in pan-methyl blot. Now, if this is true, where does this get methylated? We have done peptide matching. We have found a couple of places where lamin B1 get methylated. Today, I'm going to show you one methylation which we have worked out in this study. Here we have a wild type lamin B1, which is tagged with wasabi, which fluoresces green. When we express this, again, as you can see, it goes to the nuclear periphery. Some amount of it is present in the plasma as well. We also labeled this, performed immunostaining with the endogenous lamin B1, and you pretty much see the same profile. What we have done is we have created a mutant where we know that this lamin B gets methylated, which is a lysine at 417th residue of lamin B1. When we express the mutant protein of lamin B1, what you can see is hopefully here, lot of accumulation of lamin B1 in form of these little circles or speckles. If I leave this nucleus for a little longer, all the peripheral lamin B1, which you can see, will bleach out into this nucleoplasm and eventually move to the cytoplasm, distorting the nucleus very badly. So we know with this experiment that methylation deficient mutant of lamin B1, while it has capability to go to the nuclear periphery, it is not stable at nuclear periphery. So EHMT1 mediated methylation of lamin B1 is essential for maintaining the peripheral localization of lamin B1. We have looked at lamin AC status in these cells, and when mutant lamin B1 is expressed, we realize that lamin AC also shows a pattern which is matching to lamin B1, which is a mutant lamin B1, which indicates that that is dominant negative in nature. Now, if that is the case, what will be the nucleus, how will be the nucleus of this mutant lamin B expressing cells look like? Here is a nucleus where wild type lamin B1 is expressed. Again, you can see a beautiful nuclear membrane underneath which there is peripheral heterochromatin. But the moment I express the mutant lamin B1, what you can see here is peripheral heterochromatin is almost vanished from here. There is no peripheral heterochromatin in this. So this is a very striking phenotype, but we believe that this is because of the expression of the dominant negative form. But it corroborates with to a certain extent with what we have seen with loss of EHMTs as well. So basically, mutant lamin B1 is not stable at the periphery, and it acts as a dominant negative for lamin A as well, and it results into a complete loss of peripheral heterochromatin organization. Now, to test this, indeed, if lamin B1 is methylated in in the natural context also, we performed the same experiment where we immunoprecipitated lamin B1 from different age groups. This has to say fetal, somehow the labeling is messed up. Immunoprecipitation of the lamin B, where you can see that it is immunoprecipitated in all 3 age groups here. The levels have declined because I told you that overall lamin B levels go down. When you look at the methylation signal in here, in these cells, you can see that fetal and 31 years old cell have a nice signal of the methylation, but at 65 years, while there is some amount of the lamin B1 still there, the signal is near complete lost, which indicates that in the context of aging, methylated lamin B1 is substantially reduced, and this could be one of the reasons for instability of the lamin B1 at the periphery and eventually its loss, which leads to a loss of peripheral heterochromatin also. Now, if all this were to be true, I should be able to rescue this phenotype in aged cells. We precisely did that same experiment where we have taken aged cells. Again, you can see here, there is complete absence of peripheral heterochromatin while nuclear membrane is beautifully present here. We overexpressed our constructs EHMT1 and EHMT2 in these cells. And what you can see is overexpressed cells show beautiful recovery of peripheral heterochromatin organization. What I'm not showing you today is overexpression of EHMT1 actually brings back the levels of lamin B1 also. I told you it regulates to a certain extent via transcription also. That's what we tend to see here. So over what I told you today is loss of EHMT1 results in loss of lamin B1. It influences the stability of the lamin B1 at the nuclear periphery, because of which there seems to be a loss of peripheral heterochromatin. I can rescue this effect in aged cells by putting back the gene of interest. This is what, in summary, I have told you. I'll take you to some observations recently because I promised you that I'll talk to you about aging. This was the story of aging. I'll talk to you about little bit about what happens in pluripotent cells also during the course of reprogramming as well, right? So the way we started this study was we wanted to make human-induced pluripotent cells knocking down EHMT, and we really couldn't make iPSCs because the efficiency was extremely low and the cells were showing the senescence phenotype. So the whole story came because of that. Now we know that loss of EHMTs act as a barrier for reprogramming. So then how do we still wanted to study what are the contributions of EHMT1 in reprogramming? So how do we go about it? I'll tell you in next 2 slides how do we go about it and what we have learned from it. So just to discuss with you briefly how cellular reprogramming works, it's not new. Everyone else knows about how cell reprogramming works. Here are the fibroblasts, which we have used in the previous study. These are primary fibroblasts. When you put Yamanaka factors to these cells, they start to undergo this process called as mesenchymal to epithelial transition, and you tend to see these cuboidal cells arising in the culture. When you let these cells grow in the culture under appropriate conditions, they make these kind of the colonies, which are iPSCs or pluripotent. They are very, very similar to ESCs. Now, why should we do this? And what are the parallels between what I showed you in terms of aging, right? Aging is one extreme. If you consider development in the center, aging is a far extreme when you are going down the hill. But if you think of reprogramming, it is rejuvenation, right? You are going from a differentiated state to an undifferentiated state, another spectrum. So this would beautifully allow us to model what are the events that might happen and how lamins and EHMTs might contribute in the process of aging. I discussed what might be its role in the context of rejuvenation. So before I go to that, I'll tell you how the cell nucleus of a pluripotent and a differentiated cell looks like. You can see here in yellow, this is a differentiated cell, which we have been talking about, the fibroblast cell. I just want to highlight few things. There are plenty of things written here. Please don't look at that. Focus on what I'm trying to show it to you. A differentiated cell has both the types of lamins, all three types of lamins for that matter, lamin A, C, and lamin B, which gives the rigidity to the nucleus, right? They are present beautifully, and peripheral heterochromatin is nicely anchored onto this. In addition to anchoring of the peripheral heterochromatin, there are some unique nuclear membrane proteins which are present in a differentiated cell versus the pluripotent cells, which we will talk in a minute. Now let's look at the pluripotent cell. Pluripotent cells do not have lamin AC. ESCs do not express lamin AC. They only come up during the differentiation. So basically, the lamina is made up of B type of lamins. I was telling you the loss of B type of the lamins is not tolerated. That's why people believe that you don't tend to see any mutations in B type of lamins. So the composition of the nuclear membrane proteins is also different in the pluripotent cell to a certain extent. If you talk about the amount of the peripheral heterochromatin which is present at the nuclear periphery, earlier it was thought that there was absolutely no peripheral heterochromatin which is present, but now it's not true. There is some amount of the peripheral heterochromatin is present. The amount is quite low compared to the differentiated cells. Okay, so now when we talk about reprogramming from a fibroblast cell towards a pluripotent cell, basically we are crossing this boundary from yellow to green. It means that the composition of the nuclear lamina has to change, the nuclear membrane has to change, the tethering capabilities of of heterochromatin also has to change. So when does this happen? I'll show you in just 2 slides. It's unpublished data. We are still pursuing. Quite a bit of information is available, but we are in some fag end of this story. So I'll just give you some briefs highlights about this. So when does the composition of nuclear periphery changes during the reprogramming? So towards that, we used approach of imaging because the number of cells which are undergoing reprogramming are very less. So doing biochemistry becomes a bit difficult here. So we started our experiments with imaging studies. Now you can see here, I told you the intermediate state is a mesenchymal to epithelial state, which I discussed with you. Here is a fibroblast nucleus where you can see lamin B is present beautifully at the periphery and in the nucleoplasm. I hope what you will appreciate here is in pluripotent cells, may it be iPSCs or ESCs, the pattern of lamin B is strikingly different. You tend to see more of this exclusively at periphery. There is some at plasm as well, but the ratios are completely different here. Most of it is present at the nuclear periphery. I would also want you to notice here, during the intermediate stages where some cells are now undergoing reprogramming, you can see the localization of the lamin B1 is slowly shifting towards the periphery. The cells where you can see it's present both in the plasm as well as the periphery are the fibroblast cells, and here are the stacked up colonies. These are cuboidal cells. They attach to each other and they grow on each other. So you can clearly make out that these are the cells in the colony. Now they have shifted their expression profile from plasm to periphery, which is beautifully now present in the pluripotent cells. This is about lamin B1. If you look at lamin AC, again, the profile of lamin AC looks more or less similar to lamin B1. The levels of the lamin B1 slowly starts to change. You can see that, and drastically reduces in PSCs. I told you the composition of the nuclear membrane is also different in pluripotent cells versus the differentiated cells. I'll show you one such membrane protein, which is lamin B receptor. You can see that fibroblasts have almost negligible levels of lamin B receptor. This is highly exposed image just to see that, do we see anything at all? It's really negligible. Lamin B receptor is almost not there in terminally differentiated fibroblast cells, but they start to acquire or its expression in intermediate state. Where you can see its localization everywhere, periphery as well as in the plasm. And now by the time it becomes stable and the iPSC line is formed or the cells acquire pluripotency, you will see that its localization is similar to what is of lamin B1. So during pluripotent reprogramming from fibroblast to pluripotent cells, this mesenchymal to epithelial transition does a lot of change in terms of nuclear lamina as well as nuclear membrane proteins. There is a unique way of presenting its lamin exclusively at periphery. Certain lamins are lost, and new kind of nuclear membrane proteins are now present in these cells. So this is how the nuclear lamina is remodeled during the process of cellular reprogramming. Beautiful. More or less this is expected because we know fibroblast, we know ESC. What we have figured out is at an intermediate state. Now what happens to EHMTs during this process? Now this is what happens. We have done high-end imaging, sim imaging for this to show you what I'm showing you now. Here is a fibroblast cell. You can, red is lamin. You can see that lamin is present at the periphery of the nucleus. Green is EHMT1, which is present in the nucleoplasm. Very few yellow dots you will see at the periphery. Basically, you have to squint your eyes to see that it is at the periphery. I'll show you a colony of pluripotent cells. Now, what you will appreciate is euchromatic histone methyltransferase 1, which is predominantly a nucleoplasmic protein, which we have seen so far in the fibroblast, is now suddenly a part of it has really moved to the periphery of the nucleus, and it co-localizes with lamin B1 at the nuclear periphery. Now, when does this happen? This happens at the intermediate stage where we have started to express OSKM and the transition is happening. Now, I hope you can see that good amount of the protein is now being deposited at the periphery. These are antibody stainings, guys. So lot of, for some reason, we tend to see a bit of a background with the lamin B1 antibody only with the differentiated cells, while it shows beautiful staining for us in the PSCs. So this was a striking thing, right? A chromatin modifier, which methylates lamin B1, we know that it does it in fibroblasts, doesn't really has to localize to the nuclear periphery. We really don't know where does it methylate and how methylated lamin B moves. So, but seems like in pluripotent cell, the enzyme, the entire machinery seems to move itself to the nuclear periphery. We are still trying to figure out that what is the significance of this? Why do you need a chromatin modifier, a methyltransferase, which can do its job, seems anywhere, as evident from our fibroblast cell, has to move to the nuclear periphery? There is one just guess for us. Lamin B seems to be in a constant flux in PSCs. So we believe that because it's the only lamina which is present and has to be maintained at the periphery, the enzyme complexes are moved there so that the turnover and the stability of the protein is maintained. While we are working on that, I really don't have any concrete experiment to tell you at this point of the time. Another one data set I will show you, last data set, is if the enzyme itself is localizing with lamin B1 at the nuclear periphery, so then what is happening to the methylation state of lamin B1? I hope you can appreciate that in fibroblast cells, we have some level of lamin methylation, which are low. But the moment we start to express OSKM, we tend to see these methylation levels increase. But in PSCs, we tend to see the highest level of methylation of lamin B1. So EHMT1 seems to have non-histone role as well during the process of reprogramming to induce rejuvenation towards this differentiated cell, which is what is an active project going on in the lab. And hopefully I'll be able to share more about this in future. These are our observations. EHMT1 localizes to the nuclear periphery in the pluripotent cell. We detect enhanced lamin B methylation in these cells. What happens to chromatin tethering? Why there is not much of the chromatin tethering, even lamin B1 is stable there, etc. All those questions we are addressing in the lab right now. But I'll end with this. And before that, I just want to thank all my students who have done this work. Kriti, Alhad, and Radhika are the key players. Alhad and Radhika has done aging part. Kriti is now pursuing the reprogramming part in the lab. They have been a fantastic bunch of the students, very hardworking and very cooperative with each other in working in a group project like this. I have fantastic collaborators from INSTEM. Das and Akash have been great and supported a lot of data analysis in terms of high throughput information imaging, etc., both the labs. Chandra Prakash Chaturvedi has provided us with the plasmids, which we have used for overexpression. Marjorie has been a fantastic collaborator where we have been able to do mass spectrometry and other experiments. Thanks. We had a great facilities at INSTEM where I have been for past couple of years. Sorry. Lab has been funded by India Alliance funding for now it is over, but when I started my lab, I started with this funding. Now we are working currently with the SERB and DST grants. And I would like to thank all of you for coming on a Sunday morning to listen to what we do in the lab. Thank you.
Thank you so much, Shravanti, for such an interesting talk on the role of methyltransferases. And it's very interesting to see the changes in the nuclear composition during reprogramming. Beautiful imaging, I must say. So we've received a lot of questions, but on account of time, I have to cut short some of them. But I'll able to take some of these questions if you allow me to.
Yeah.
So the question is from Vani. Do lamins disassemble and reassemble during reprogramming? And what drives their shift in localization? I mean, what takes them to the periphery?
Yeah, so that is what we have been debating about. We know that the form of the lamin, or at least in literature, we know that the form of the lamin which is present in the plasm is more of the depolymerized form. And again, I'm speaking from the perspective of what I know from the literature. We are conducting those experiments in the lab now to see whether it's a polymerization, depolymerization event, or there is really loss of lamin and there are fresh lamins which have been made. We really don't know. It is a recirculation or making it from scratch, because usually lamins are very long-lived proteins in a differentiated cell. The turnover is pretty slow. So we don't know during the course of rejuvenation, does the cell opts for recirculating it and keeping a part of it, or it just starts from the scratch? Those questions needs to be answered. No idea.
Right. I'll take one more question. This is from Deepak Korea. Do you know which lysines on lamin B1 are methylated? If you replace the lysines with leucine or methionine, are they able to sustain nuclear organization or heterochromatin localization over aging?
Yeah, so I showed you about one methylation mutant, 417 methylation mutant, which actually takes it to the periphery, but somehow it doesn't stabilize there. It comes out eventually. We have another two positions where we have seen that lamin gets methylated, which we have not published yet. It's 219th or 17th position. I just somewhere there. There are 2 residues which we have found that which are critical for maintaining the stability of the lamin at the periphery. Yes, it does. Yeah.
Right. Okay, I think there is still a lot of questions out there. I don't think we'll be able to cover all of them in the short period of time, but I hope you'll be able to stay with us during the rest of the session. And if you don't mind answering some of them in the chat box, it is there. So that will be very helpful.
Yes.
Thank you once again for answering all the questions, and thank you for taking the time to speak in this conference on a Sunday morning in India.
Thank you. Thank you for the invitation. It was a great pleasure.
Thank you very much.
Bye. Bye.
So moving on to the next speaker of this session is a physician scientist, Dr. Karishma Kaushik, who is currently an assistant professor at Institute of Bioinformatics and Biotechnology at Savitribai Phule, Pune University. She did her MBBS in 2005 from Dr. D.Y. Patil Medical College, Pune, and a MD in clinical microbiology from Armed Forces Medical College, Pune. Thereafter, she joined University of Texas, Austin, USA, to do her PhD and was later appointed as an assistant professor there until 2018, when she decided to move back to India to start a new lab in Pune University. She has received numerous prestigious awards like the Ramalinga Swami Re-Entry Fellowship from DBT India in 2018 and the Innovative Young Biotechnologist Award from DBT in 2020. She is actively involved in science outreach activities and has also contributed to many popular science articles. I was intrigued by reading one of her articles in Nature India saying, "What works in mice may not work in humans, especially in infectious disease," arguing the need to generate human relevant models. And on the same line, Dr. Kaushik will be delivering her lecture today. So without further ado, we welcome Dr. Karishma Kaushik to Biologically Speaking, and looking forward to your talk.
Hello, everyone. Can you hear me?
Yes.
Yes. And Sonak, actually, I'm trying to share my screen before I get ahead. I have sent you an email. Can you just check in case I'm unable to share it? You can share it from there. Yeah.
Sure.
Sorry for these technical glitches.
I got it. Yeah.
We always start sharing the screen only 1 minute before the talk, you know. So this is very last minute always. So once so
I think you are co-host, so you should be able to.
Yeah, but for some reason I'm unable to. I don't know what it says, you know, system preferences need to be changed or something. And I'm not sure if we want to do it right now.
Okay. I mean,
can you share?
I actually didn't get the presentation file.
Okay. Just a minute. I'm really sorry about this, but I have sent it to you.
I got your email, but there is no attachment.
Just a minute. Just a minute.
I mean, yeah, no problem. I mean, we can retry again in case there is something.
Yeah. All right. Or 1 minute. I can just join from another system where maybe it will work. Okay. I'm mailing it to you again.
Okay.
Yeah. I have sent it. Did you get it?
No, I've still not received it. Oh, yeah, I got it now.
Okay. Okay. So will you be able to share it? In the meantime, can I get started talking?
Yeah, let me see where it's. Yeah, I think it's downloading, so it should be fine.
All right.
Yeah, I think it's downloaded. It's taking some time, but it should be fine, I guess. You can start. I'll share it in the meantime.
All right. Okay. So at the outset, I think I'll use this as an opportunity to take some time to say thank you very much for this invitation to Biologically Speaking, HerStory of Science organizers, Bias Watch, and all of you put this together. This means a lot to me on a couple of fronts. For one thing is to be getting an opportunity to talk with people at a conference where you're showcasing the work of women researchers is, it's particularly an honor. And secondly, the fact that I would say an early career woman investigator in India, who's recently moved back in 2018 to start my group. I was thrilled, excited, and very eager to accept the invitation when I received it. So thank you, Sonak. That was wonderful. Very good to see you all here. I know I've interacted with several of you through Twitter. And that's been really good, especially in the time of the pandemic where, moving out of the house has been a challenge. Sonak, is everything okay?
Yeah, I'm still trying to do it.
Yeah,
I'm
that's
sorry.
okay. No worries. I can take my time, and I have only a 15-20 minute talk, and there'll be room for questions. So no worries about that. It's a pretty short talk.
Yes, I think it should work now.
Yeah. Just let me know.
Can you see it now?
Yes, absolutely. Thank you, Sonak. Thank you.
No
And
problem.
sorry
No
for the pressure.
No problem.
All right, great. Thank you so much. So this is who I am. I'm Karishma. I'm a Ramalinga Swami Fellow at the University of Pune. As Sonak said, I moved back to India in 2018 to start my group on the Ramalinga Swami Fellowship. And it has been an exhilarating professional journey ever since. And I hope to give you a sampling of this through my talk. And we can, of course, take this offline through email and Twitter anytime. I'm always excited to interact with the community at large in any way possible. So like Sonak said, I'm a physician scientist. We can move to the next slide, Sonak. Thank you. And I hope today to talk a little bit about, to give you an appreciation and understanding of this entity known as biofilms. I gather from the 2.5 years I've been in India that there are not too many people working on this, which is a good thing and a bad thing. We'll see how it goes in the long run. I hope to give you an appreciation and understanding of biofilms, and also I hope to convince you that we are trying to fill a gap that exists and the mode we are using to fill this gap. All right, so let's zoom out. All of us have had a wound at some time in our life. Any lucky souls who've never had a wound? I doubt that, right? If you go way back in time, childhood, teenage years, a wound, I mean a physical wound, a cutaneous skin wound, not talking of the other variety. All right, so we've had a cutaneous wound at some point in our life when we've fallen down. It's defined as a breach in the skin and the underlying tissue. And most often than not, we bleed a little, it stops bleeding, and then we spend maybe a week, a little more than that, seeing it heal. So you see there is formation of tissue, and eventually it just, it's left behind as a scar. However, there are large groups of people, and this is an ever-increasing population, where these wounds don't follow this orchestrated pattern of healing, but instead get arrested at different stages. We'll talk about what those stages are. But this group of people include people with comorbid conditions like diabetes, hypertension, lack of physical exercise, sedentary lifestyle, and in these groups of people, wounds can take a chronic non-healing form. And this, most people working on wounds would consider as a silent pandemic. And I know pandemic has extraordinary proportions that it takes when you use the word today, but this is a silent global pandemic which is draining the healthcare system and contributing hugely to morbidity among groups of people where wounds don't heal. All right, we can move to the next slide. So in what stage of healing does this wound get arrested in a chronic non-healing wound? Well, it happens in that stage of granulation tissue, or the inflammation to proliferation phase, where your wound has a gap and now your body is trying to fill it by proliferating new wound bed cells, which happen to be the skin cells, fibroblasts and keratinocytes. So that's when you see this pale pink, salmon pink, fleshy thing growing in the defect in your skin. And that's granulation tissue. So what happens to granulation tissue, which leads to this non-healing path versus the predictable healing path? So there's a widespread consensus. We can go to the next slide. The widespread consensus is that formation of biofilms in granulation tissue, in the wound bed, is the single most important cause of non-healing wounds. And what are biofilms? Biofilms are microbes, right? There's presence of microbial infection. However, biofilms are these three-dimensional, highly structured organized communities of microbes, and not just a physical consortium, but there is intercellular communication. And there are emergent properties that this consortium confers to the commune as a whole. So it's believed that in the wound bed, formation of biofilms is the most important cause of non-healing wounds. And biofilms appear like this. So if you see the blue nuclei are your DAPI stain of host cells. And what you see is the red here and green here, which is the clumps of bacteria, which are biofilms. Most important bacterial pathogens contributing to this non-healing state by forming biofilms are Pseudomonas aeruginosa and Staphylococcus aureus. It's important to note that biofilms are most often polymicrobial as well, meaning that there are often two, if not more than two, species of microbes, maybe bacterial plus fungal, existing in the wound bed. So it's a mixed species consortium that leads to this protracted state of inflammation and non-healing. Okay, we can go to the next slide. So as you can see, I get very excited when I talk about biofilms. They are close to my heart, close to my work. So obviously biofilms don't exist in isolation. They are communities, however, they exist in close approximation with the host environment. And what is this host environment in the wound? It's the host cells, the fibroblasts, the keratinocytes. It's the matrix elements, the collagen, fibronectin, fibrinogen. It's all the biochemical factors that are secreted, lactoferrin, lactic acid, matrix metalloproteinases. And there is an additional layer of complexity if you think of this interfacing with the immune compartment, with immune cells through the capillaries, all right? So the take-home message is biofilms exist in approximation with the host environment. We can go to the next slide. And this interaction is far from static. In fact, it's a hugely dynamic interaction where biofilms influence host components and host components in turn shape the properties of the biofilm. So, I mean, the bottom line is that there is this microenvironment which is hugely active, dynamic, and constantly changing. And it's in this milieu, we'd call this conglomeration of host factors a milieu. It's in this milieu that biofilms exist, thrive, and do all the things that we don't want them to do. And what are those things? We can move on. So it so happens that biofilms are notoriously recalcitrant to antibiotic treatments. There are different reasons for this, which account for their physical structure, like they impede the movement of antibiotics. They also adopt metabolically dormant forms of bacterial cells, which may be less susceptible to antibiotic effects. However, the end result is that when treating a chronic non-healing wound, you're throwing everything at the wound, including the bath water and the kitchen sink water and everything. So chronic wounds usually just get a bombardment of antibiotic treatments. Oftentimes they don't heal in spite of these various treatments, resulting in even prolonged treatment regimens. Why they don't heal is a mystery. Why they heal is equally uncertain. And so we really don't have a targeted, precise mechanism to deal with a non-healing state. Okay, we can move to the next slide. So lots of work has been done in the lab by developing novel approaches moving beyond conventional antibiotics like nanoparticles, blue light therapy, hyperbaric oxygen therapy, modulation of pH, all of which are known to influence the susceptibility of the biofilm to antimicrobial treatment. So used with antimicrobial treatments or used alone. However, what the issue with all of this is they show excellent results in the lab. All right, it's wonderful. However, when we take them further down that pipeline of analysis of a compound, we go to further chemical studies and then we go to actual model systems, we find there is a huge attrition, there is a huge fall. So we spend so much time in this early preclinical stage of trying to understand what the compound is, what it does. We get exciting results only for it to fall when it reaches a more relevant model system. So one of the ideas is if we have all these different compounds, maybe we can adopt an approach where we say, let us test more and let's fail fast. All right, let's reach as many new compounds that are being developed to overcome biofilms in wounds, and let's fail fast so we can move on to the next compound and not waste more time if it's not going to work. So where is this cliff? Where is this fall happening when you have thousands of lead compounds and then very few reach the other end? 1 in possibly 50, they say, for biofilms, which is actually an optimistic estimate. So where is this cliff? We can move to the next slide. The cliff happens to be the wound biofilm systems. So how is the community? How is my community studying wound biofilms so far? Well, we rely on in vivo, those who have the facilities, those who can afford it, and those who have the expertise. or ex vivo, or then we have in vitro platforms, which I'll make a case to you are highly reductionist. So if you go to the next slide and you look at in vivo systems, you have lots of small animals. We have mice, we have rodents, and we have fewer larger animals like porcine systems. I don't need to tell you what the limitations of these are. Scientifically, I can tell you one thing, that mice do not heal wounds the way humans do. Mice heal wounds by contraction, by pulling together the edges of the wound, whereas as I just told you in detail, humans heal wounds by proliferating the gap to fill it. So there is a fundamental difference in which mice do it and humans do it. Apart from this, we have technical concerns with infecting mice, especially in facilities not capable of infecting small animals. There are costs and there are huge ethical implications. Needless to say, these are not high throughput systems. So in vivo systems have scientific and a plethora of other limitations. Let's look at ex vivo systems, all right? The next slide. So what if we took a piece of skin out from the pig, which is supposed to show wound healing very close to humans? Of course, rearing pigs in the lab is a different story altogether. Or we take it from human skin, like discarded cosmetic surgeries or discarded procedures, and set it up in a transwell platform, add in elements. What are the limitations? Firstly, technically, this is tough. This is tough to execute, to keep ex vivo skin alive in a petri dish. Secondly, it's a static system, right? We are not really accounting for many factors that play a role in the wound infection microenvironment. Okay, so ex vivo, to be honest, I currently in my facilities would find ex vivo systems a huge challenge. Okay, so following on, we say, all right, if we can't do in vivo, ex vivo, can we do in vitro? Yes, it's possible. However, the in vitro models that exist are highly reductionist. They are highly simplistic. So they either just shaking cultures or drip reactors or microfluidics, which are hard to fabricate. So while people have attempted to include host elements like blood, plasma, some matrix factors, the current models that exist aren't very holistic. They aren't very representative of the wound microenvironment as a whole. All right. Okay. So moving on to the next slide, we believe that there is a gap that exists between acknowledging that chronic wound biofilms are a problem, developing models to study them, and developing tractable human relevant models. And this project that I'm going to talk about is about filling this gap that exists in the community. All right. So what is this gap? Let's move on. So the gap is, how do we plan to fill this gap? So the idea is, can we build an in vitro platform that doesn't have the challenges of in vivo and ex vivo systems, and yet is much more representative of the clinical wound microenvironment in the context of biofilms as compared with in vitro reductionist platforms? So can we somehow bridge this gap, pun intended? And what would this do for the wound biofilm community? This would provide us a high throughput tractable platform, hopefully, to test therapeutics. So that test more, fail fast approach, right? Because it's high throughput. It would give us some insights which are relevant to host pathogen interactions in the clinical wound bed. And we could also leverage it to complex platforms like microfluidics and transwells by including this in those platforms, including this as the human relevant milieu. All right. For beyond wound biofilms, this would serve as an alternative to animals being used, and it would enable some precision-based, personalized antimicrobial therapeutic development for wound biofilms. So how did we go about this? We said, okay, so far people have worked with LB broth, which all of you are familiar with. It's a highly protein-rich broth. I don't think the wound is bathed in LB, not the last time I checked. People say, all right, if not LB, let's do serum. But I made a case that saying the wound microenvironment is much more than just serum. It's a host of other factors. So we said, let's compare what are clinical reports of wound milieu or wound fluid composition. And we looked at lots of clinical publications and concluded that 70% serum would recapitulate most of the biochemical factors present in the wound milieu. In addition to the factors that were, so to account for the factors that were lacking, we said we will add matrix elements like collagen, fibrinogen, fibronectin, and two important biochemical factors, lactic acid and lactoferrin, both are which seem to be increased in non-healing wounds in the presence of infection. So we developed an in-house proprietary wound milieu formulation, which we call our in vitro wound milieu. And our subsequent studies have used this in comparison with Luria Bertini broth and fetal bovine serum alone to understand whether this can be filling that gap between complex in vivo and reductionist in vitro. So let me take you on this journey. Let's see what we found. Okay. So the first thing we did is, after making the milieu, we said, can we even grow anything in this? Is anything going to grow in this? So we took our wound relevant pathogens, Pseudomonas and Staph, and we just did planktonic growth curves. So if you see in LB, both of them grow really well, not Nobel Prize winning research. Everyone knows Pseudomonas and Staph grow in LB. Then we looked at serum alone. And we found that while Pseudomonas grows pretty well, Staph appears to be retarded in growth. This is only serum. And then we took our wound milieu and found Pseudomonas grew actually very well in the milieu and Staph grew poorly. So the milieu, our in vitro formulation, was exaggerating the effect of FBS, where it's providing a clear advantage to one pathogen, which is Pseudomonas, and not the other, which is Staph. And these are the two bacteria grown alone. They're grown alone in this medium. This is interesting because in vivo clinical reports also suggest that though the two pathogens coexist, Pseudomonas appears to outgrow Staph. However, this was alone, this was individual. Let's look at if we grow these two pathogens together in the milieu. Here we are going to actually parse the role of interspecies interactions. Okay, so this is alone, these initial graphs, and subsequent, the last two are co-species. And you clearly see, all right, when alone they grow fine, Pseudomonas appears to grow better than Staph. But when they are growing together, you see Pseudomonas has a slight growth advantage. It's growing better, and Staph is growing poorly. All right, so we clearly see even when grown together, Pseudomonas is outcompeting Staph. So this adds on to the growth advantage we saw in the earlier slide when they grow alone. And this can be seen very clearly here when you look at a ratio of colony units, co-species versus monospecies. And I know everything collapses into one graph, but the young researchers who worked on this are going to be looking at it and saying, oh, imagine the number of plates we put up to count these colony forming units. So, all right, LB, both get inhibited. In serum, Pseudomonas appears to have a slight advantage, whereas in wound milieu, of course, it's an exaggerated advantage under co-species conditions, even when grown together. So clearly, Pseudomonas outcompetes Staph in the wound milieu, and this is in accordance with clinical observations. Okay, then we said this was planktonic, but I've made a whole, planktonic is these free living cells, right? Free floating cells. But I've made this whole case to you that wounds are about biofilms. So can the wound milieu support the growth of biofilms? And the answer is yes. Importantly, for Pseudomonas and Staph, they support the growth of biofilms, and these biofilms are metabolically active, meaning they contain a significant proportion of living cells, and this is assayed as a proxy. Maybe Sonak, you can move to the next slide. It will show up the, yeah, thank you. This is, no, the earlier one, yeah. And this is shows up as a proxy where the living cells in the biofilm convert a compound formazan to orange color, right? So the orange color becomes the proxy for living cells, more orange color, more living cells. It's a rough estimate. This is similar to the MTT assay that many people working with cells, human cells, may be doing, mammalian cells. So the XTT assay told us that wound milieu supports the growth of biofilms, and the biofilms are metabolically active even when grown together, even under co-species conditions. However, we don't know which is contributing a major component of the metabolically active part, Staph or Pseudomonas, and we look at that subsequently when we image the biofilm. All right. So it seemed to tick off the next thing on the list. It was supporting metabolically active biofilm growth. We can go to the next slide. But I also told you that wound biofilms are notoriously resistant to antibiotic treatments. We call them tolerants, actually, in biofilm parlance. And if you look at the wound milieu, you find that compared to LB, which is your standard media widely used, Pseudomonas is 8 times more tolerant to the effects of tobramycin, a commonly used antibiotic. And Staph is also highly tolerant to the effects of vancomycin. And this is notable in comparison with serum. So while we notice that the effects with serum and wound milieu are not very different, you can go to the next slide. Yeah. While we notice they're not very different, the earlier slide. Yeah, the value being the same, 8 microgram per ml, probably underscores the role of a dominant role of serum in conferring antibiotic tolerance to Pseudomonas. And the high tolerance we see with Staph aureus to vancomycin underscores the role of matrix factors because it was highly susceptible in serum alone, which when we added matrix factors and those biochemical components, we suddenly saw an increase in tolerance. So by and large, the wound milieu recapitulates the tolerance effects which we see for the two pathogens with commonly used antibiotics. All right, and this was a modification of MIC. It's called biofilm eradication concentration and defined as 80% eradication effects. Okay, so all right, so it recapitulates the tolerance. Now what happens when, and this was a big question we were all itching to answer, what happens when you actually see the biofilm under the microscope, under these different conditions? So we have fluorescently tagged Pseudomonas and staph with GFP, Pseudomonas with M. cherry. And if you look at Staphylococcus aureus, you see these beautiful dense mats. I would say that in the wound, we really don't see them as these dense mats. They're seen as clumps. Nevertheless, this is in a black 96 well plate compatible with confocal microscopy. And the imaging we've done here is tile scan. So it literally scans the entire biomass in tiles and stitches it together. So each of these wells can sometimes take up to 40-45 minutes to image. So in LB, you get a thick biofilm. In serum, you get a thick biofilm. Not surprising, fair enough for staph. In wound milieu, you get a much thinner biofilm. This is important to note because it may mean that we are assaying compounds with these laboratory media, reductionist media. We are overestimating biofilm thickness, which means maybe we're throwing a lot of compounds to say they are ineffective when actually we're just giving them a very hard situation to work against, right? A very thick biofilm to work against. So we do see thinner biofilms in the earlier slide. Sorry. Yeah, earlier slide. Yeah, so we do see thinner biofilms in the wound milieu for staph. And when we look at Pseudomonas, we do see biofilms growing in the in vitro wound milieu as well. They're reasonably thick, about 40-50 microns. However, though we saw a lot of effects earlier in LB, we don't see them. This is because the plates we used were surface treated to reduce surface attachment of the bacteria. We wanted the role of the milieu to be parsed out. And this indicates that in the absence of robust surface attachment, the proteins, likely proteins present in the milieu, play a very important role in helping biofilm formation, which in LB are not there. So it's the host proteins or the matrix elements that are likely to play this important role in biofilm formation. Okay, all right. And when we can move on to co-species, the next slide. If you look at the co-species effects, we see a reduction in LB, that's probably Pseudomonas killing staph, as we talked about it. We see stable coexistence in serum. However, notably in wound milieu, once again, we go back to that story where Pseudomonas is outcompeting staph. You see very, very little green, very few green dots. And predominantly, it is Pseudomonas growing in the co-species condition. So our wound milieu visibly demonstrates the outcompetition, Pseudomonas outcompeting staph in biofilm co-species conditions. All right, so this is the story so far. This is similar to in vivo observations, this last piece of data as well. And if you go to the next slide, what have we shown so far, right? We have this as a preprint, and we can clearly say that we have incorporated relevant clinical components. Our platform is easy to formulate, uses widely available components that any vendor in any institute can provide you. It's tractable. We'll come to that subsequently, but widely available components. And though it uses fetal bovine serum and some animal collagen, has less ethical, technical, and cost implications as compared to in vivo systems for sure. Importantly, the platform with all its simplistic features is able to recapitulate key in vivo observations of supports co-species growth, supports outcompetition of one species over the other. It supports increased antibiotic tolerance, supports biofilms with metabolic activity. So, and it's compatible with standard biofilm assays like your XTT assays, planktonic curves, or confocal imaging. It's tractable and high throughput. So we are now trying to evaluate this platform as an early high throughput approach to screen lots of these preclinical compounds, lead compounds that are developed, and to see if it can serve as a go, no-go decision to take something further to complex systems, or to say something is unlikely to work if it hasn't worked in a system that by and large recapitulates important features of the wound microenvironment, albeit not every feature, for example, immune cells and other biochemical factors, but the important factors. All right, so I think I'll lay my case there. We can move to the next slide, where I have to say, I thank all the young women who came to my group and worked with me at a time when I was starting out. So we literally bought pipettes together. We cleaned the lab benches together. It's a matter of great pride, and particularly stated at this platform, that this is an all-woman authorship work. It's under revision. We hope to get it published very soon. Done in India, an Indian woman investigator working in India, and funded on Indian grants. So I'm indebted to the young women who believed in me at a time when I really needed it. So thank you for this platform and thank you for this opportunity. Okay, I see lots of questions, Sonak. Sorry, Sonak, you're mute. You're mute. We just switched roles for a minute there.
Yeah, no, thank you. Thank you so much for that interesting talk. And I hope I was able to coordinate with the transition.
You did.
My apologies.
I apologize for putting you in the spot the last minute. I don't know, suddenly it didn't work. I have to go to my system preferences and figure it out.
No problem. I mean, those are the plus points or minus points, what you say for the virtual media. But I'm not surprised to see all these questions that we've received here. But I think on the count of time, I'll have to take some of them for this session. And I hope you will be able to join us.
And I will reply to the rest in the chat window. Certainly.
That would be great.
Please tell me any ones you want to take.
So we'll take one from Shankalpo Venkatragavan.
Yes.
Are the microbes in the biofilms formed spatially organized? Do we understand the principles that
Great question. I mean, this is a fantastic question, Sankalp. Are you sure you're not the reviewer for this paper that we've just submitted? Because yes, I do say they are coexisting. However, we have found spatial segregation, not we, the community, other publications have found spatial segregation in the wound. So Pseudomonas appears to exist separate from staph in the clumps, often vertically segregated as well. Whether there is a single biofilm clump having both Pseudomonas and staph, it has not been shown. It has not been shown. A single clump having both, but a single wound surely has both. I hope you're understanding that slight difference. So we are not quite understanding principles that govern this, but this has come up in the reviewer saying that please make this distinction clear. So the wound has both, but does a single clump have both? That has not been shown. Okay. So like our FBS showed them in close approximation, whether like that it exists in the wound, we haven't been observed it yet.
Excellent. So I'll take another question from Smita Chaudhary. Can biofilms trigger clonal evolution of host cells from the site of infection, spread of clone cells and lead to genetic mutations?
Yes, it's well known. It is well known that biofilms serve as sources of large-scale mutant generation, antibiotic tolerant mutants, persistent mutants, metabolically variant forms. Yes, they can spread in that fashion. This is known. This is not the focus of my work, but this has been studied.
Okay. Yes. I think with that note, Dr. Kaushik, it was great listening to you. Okay. And as you can see, there are a lot of questions already in the chat box.
No problem.
But I hope you will be able to reply to them in the chat.
I will reply to them. And thank you, all of you, for the opportunity. Thank you, Sonak.
Thanks a lot. Thank you. So moving on, after such an exciting talk by Dr. Karishma Kaushik on infection and biofilms, we are now moving on to another talk on immunology and disease mechanisms. And we have Dr. Sheetal Gandotra, who is a senior scientist at CSIR, Institute of Genomics and Integrative Biology, New Delhi. Dr. Sheetal Gandotra completed her undergraduate and postgraduate degrees in biochemistry from University of Delhi and received a PhD in immunology and microbial pathogenesis from the Will Cornell Graduate School of Medical Sciences, New York, United States, followed by a postdoctoral training at Cambridge University, UK. In 2011, she started an independent group at IGIB with the funding from Wellcome Trust to DBT in the Alliance. Her research group is interested in tuberculosis pathogenesis and host metabolism, and today she will talk on lipid droplets in innate immune defense. So warm welcome to Dr. Sheetal Gandotra to this conference, and we very much look forward to your talk.
Thank you, Shaunak. Am I audible?
Yes.
Yeah? Okay. All right. Thank you, Shaunak, and thank you to the organizers of Biologically Speaking. It's a great honor to be speaking in this session. And thank you for considering me one of the, achievers in science, if that's what you think. But I think I have a long way to go. And I hope besides this 30-minute slot, I will get to speak to you about the ongoing research going on in our lab to a greater length in the future. But in the next 20 minutes or so, what I'm going to do is talk to you about one of the major interest areas in my lab, which is lipid droplets. So I think this is not gone to slide. Are you able to see
Yeah, we can see the slide, but actually
Okay.
just have to do
Yeah,
the
I
slide.
think it's just going to take a little bit of time to project in the full screen mode.
Okay.
Okay. Is it good now?
I think it will still come here. Yeah, perfect. But
Okay.
it's coming as the presenter mode, I guess. So we can see two slides together.
Okay, let me fix that. Can someone help me with how to close this view and go to the
I think you can
presenter?
do the slideshow. I guess that should work.
Sorry about this.
You can escape that, and then at the bottom you can see.
Sorry. Just give me a minute.
Sure.
Is this still in the?
We can see all the slides here. I think at the bottom you can click the slideshow.
This should be better.
Let's say it's coming. Yeah, perfect. It's working perfectly fine. Oh no, it's again gone back.
Okay. This should work now.
Yes.
Yeah. Okay.
We
Great.
can see it.
Okay.
Perfect.
Sorry about this delay.
No problem.
Okay, so what we study in the lab are these organelles called lipid droplets, and we study them in the context of the innate immune defense against pathogens, and the pathogen that we are interested in is Mycobacterium tuberculosis. So as you're looking at the screen right now, in red are these organelles that are called lipid droplets. They store excess fatty acids in the form of neutral lipids like cholesterol esters and triglycerides. And what you're seeing in green are these fat-hungry bacteria, which are basically having a feast inside this macrophage. So what are lipid droplets? So lipid droplets are organelles, as I mentioned, but they emerge from the endoplasmic reticulum membrane. So they emerge in between the two membranes or the two bilayers of the ER membrane. And the reason why they emerge there is because the enzymes that are involved in conversion of the precursors like monoacylglycerol all the way to DAG and then triacylglycerol are actually present on the ER membrane. So the key enzyme in this process is the enzyme DGAT1, which will come up later on in my talk as well. It's an enzyme that converts diacylglycerol to triacylglycerol, which is an extremely neutral lipid, which forms this sort of lens in the middle of or between the two bilayers, the two layers of the bilayer membrane. And at some point, because of asymmetric distribution of phospholipids and proteins on the cytoplasmic versus the luminal face of the ER membrane, these lipid droplets emerge as independent entities away from the ER membrane. And these are not just bags of fat floating around in the cell, but they're actually covered by specific proteins which allow them to engage with other functions within the cell. So some of these functions that I want to highlight before I move on to host-pathogen interaction is, for example, the interaction of lipid droplets with mitochondria. So as I said, the lipid droplets are housing excess fatty acids in the form of neutral lipids, but these stores are meant so that they can be utilized in cases of starvation, for example. So the fatty acid that's released from the lipid droplet must then enter the mitochondria for beta oxidation to take place. And this is a regulated process as well. Lipid droplets can also undergo degradation by means of interacting with structures or organelles like lysosomes, and there are processes whereby this happens in a chaperone-mediated and a direct lysosome-based autophagy process, wherein the lipid droplets get basically this vectorial transfer of lipids from the lipid droplet to the lysosome for degradation. In addition to these functions, which are known to take place in several cell types, the importance of lipid droplets in the immune cells like mast cells, eosinophils, dendritic cells, and macrophages has been appreciated as a potential source of lipid mediators of signaling. And particularly in immune cells, these lipids called eicosanoids, which are generated by oxidation of the fatty acid arachidonic acid. But besides this, lipids like diacylglycerol, monoacylglycerol, PUFAs, resolvins, ether lipids. And what's interesting is that several of the enzymes that are involved in the degradation of lipids to generate these products are localizing to the surface of the lipid droplet. So with that in place, what I'd like to move now towards is the idea that these lipid droplets can also be engaged in this host pathogen crosstalk. So there are several examples out there. I just like to point out a few here. So for example, Chlamydia trachomatis, which is a cellular parasite, makes what I call these inclusion within the host cell that is that there are proteins of chlamydia that export out of the vacuole, bring lipid droplets to the vicinity of the droplet, of the vacuole, and allow this droplet to be taken up into the inclusion vesicle, and then it is utilized by the growing reticuloid bodies. RNA viruses are very interesting because they make this replication complex, which is basically a large complex of membranes on take place. Okay, and in case of several viruses like hepatitis C virus, poliovirus, there are specific proteins of the virus which interact with proteins such as DGAT1 or other proteins, that coat proteins of the lipid droplet, and allow the utilization of lipids for formation of this replication complex. Now, why this is important is because this replication complex sort of hides the genome, the RNA of the RNA viruses, away from the RNA sensing machinery within the host cytoplasm, and therefore inhibits the recognition and degradation of this viral RNA. And what's interesting is recently this mechanism has also been proposed for the survival of the coronavirus within infected cells. Now coming to my favorite class of pathogens, which is Mycobacteria species. So work has been done across various species of Mycobacteria, including avium, leprae, marinum, and tuberculosis, to find evidences where bacteria are actually closely opposed to these structures called lipid droplets. Sometimes so that the lipid droplet has been found within the phagosome, or the bacteria have been found within the lipid droplet as well. Although these are rare instances, these are all being reported in the case of Mycobacterium tuberculosis. So, given that background, just like to give you a very brief background of tuberculosis today. So while we all talk about infectious disease, and a lot of people have become experts overnight on infectious diseases thanks to the pandemic, what we don't realize is that for a disease that is curable and the leading cause of death due to any single, only 10 million new cases annually. Only I will say that because we don't have, we don't account for the large number of latently infected individuals that have a lifelong risk to develop active tuberculosis, with risk factors such as malnutrition, smoking, diabetes, and HIV infection, making them more prone to develop active disease. So it's important that we look at what are the ways by which we can target the disease and the bacteria. So to target the bacteria, there are very many antibiotic that have been used. The problem with antibiotics is that always be antibiotic resistant, right? So in this arms race between the host and the pathogen, can we look at how the host behaves such that it can contribute towards control of the infection and prevent active disease generation, or in the case of active disease, some factors that can be strengthened so that there is better healing for the patient, there is better control by the immune system itself. And so when we look at host pathogen interaction, we have to consider that this is happening in a continuum, right? So an individual, when an individual breathes in a droplet containing Mycobacterium tuberculosis, an individual may be able to control this by the action of their innate or adaptive immune system and never have any bacteria residing thereafter. Or there could be a latent infection that develops, which means that the individual doesn't actually have any overt disease symptoms, but houses the bacteria. In this scenario, the bacteria usually tend to inhabit a structure called a granuloma, which is basically an infiltration of immune cells within the infected tissue. And at some point, there is an arms race between the host and the pathogen within this granuloma, which is a structure composed of very many different types of immune cells like macrophages, dendritic cells, lymphocytes, neutrophils, eosinophils, and mast cells as well. And at some point, the individual converts from a latently TB infected individual to an individual with subclinical TB disease, and then into an active TB disease. So the host factors that play a role in controlling disease progression at each of these stages need to be understood. What my lab is really interested in understanding is this host pathogen interaction that takes place in an active TB disease. And we appreciate the fact that the structure that I mentioned, the granuloma, is not a unique identity. It is not a single identity, let me put it that way. So there is a spectrum of different types of granulomas that can exist even within the same individual. So for example, a nascent granuloma, you will have, as I mentioned, a accumulation of macrophages, neutrophils, lymphocytes, and the bacteria in this case would be mostly intracellular within macrophages. However, in actively infected individuals, especially individuals who have hard there is an evidence of cases that there is a lot of extracellular bacteria within the center necrotic region of the granuloma, and then there are macrophages just outside that central necrotic core, which also harbor Mycobacterium tuberculosis. So we know that Mycobacterium tuberculosis is a master manipulator, and that's how it's able to survive all the challenges that the host macrophage throws at it. So for example, we know that lysosomes are meant to degrade incoming pathogens, and Mycobacterium tuberculosis is able to evade the phagolysosome fusion machinery. And even if it is able to enter a low acidic environment, it's actually able to prevent acidification of its cytoplasm. So it resists it. there are processes by which it can actually escape from this phagolysosome compartment by permeabilizing it. And typically what happens is there are recycling systems within the cell that will identify such damaged vesicles and lead to degradation of those, but the bacteria is also able to overcome those processes. So given that this is the master manipulator, how does this manipulation, what sort of manipulation takes place in the context of a granuloma that is caseous, lipid rich? So if we zoom into the schematic of a granuloma, as I mentioned earlier, you have this layer of or cuff of macrophages surrounding the necrotic center of the granuloma. And you have what are called lipid droplets in these macrophages. Now, bacteria in this macrophage can be acquiring nutrients from the lipid droplets, either by opposing directly to the lipid droplets or the lipid droplets coming into the phagosome, as I mentioned previously. We really don't understand what are the mechanisms of bacteria acquiring nutrients from the host cell. So my lab is interested in understanding lipid remodeling and trafficking in TB infection. And the work that I'm going to talk about today is work done by Neetika, Kaurav, and Dilip. And there are a large number of people on this picture right now who are also contributing to that work. Another part of my lab also works on lipid remodeling in mycobacteria itself, which I won't have any time to talk about today. So Neetika started off by asking whether MTB infection leads to mobilization of stored lipids. And for that, she took a biochemical approach. So she labeled the triglyceride pool of the macrophages. So before I start, so we use THP1 macrophages for all our work because these macrophages make lipid droplets very nicely. You don't really have to add any exogenous fatty acids to make lipid droplets in them. When they differentiate from the monocyte to the macrophage stage, you get lipid droplets. So she labeled the triglyceride pool using radioactively labeled oleic acid, which gets incorporated into the triglyceride. And then after this pulse, she chased the fate of this fatty acid, whether it could get released from the cell or not in the presence of Mycobacterium tuberculosis. So what she found is, so this is a graph showing on the y-axis the amount of free fatty acid released relative to the counts, the total radioactive counts in the cell. So what she found is that MTB infected cells released more fatty acids, suggesting that the cells were being forced to undergo lipolysis as a result of infection. Kaurav then took an approach which relied on imaging and asked whether intracellular MTB was able to utilize that stored fatty acid. So his approach was he would label the cells with a fluorescent tagged fatty acid, which had the length equivalent of a C18 oleic acid. And what we could visualize is that the lipid droplets of the macrophage would get labeled with this. He would then infect the macrophages and see whether the label got transferred to the bacteria or not. So when he did this experiment, what he found that indeed the bacteria, which is shown here in the center, this is so the bacteria in this case are GFP labeled, and in the red is the fluorescent fatty acid. And what you can see is that the fluorescent fatty acid could get transferred to the bacteria. To figure out whether this was a non-specific or a specific lipase-driven process, he took a general lipase inhibitor orlistat, and the first thing we noticed is that the lipid droplets of the cells increased in their fluorescence, which suggested that there is lipolysis happening in infected cells. And if you the of the stored triglyceride actually labeled, which means that this vectorial transfer of fatty acid from the stored lipid to the bacteria was reduced by using a lipase inhibitor. So now Dilip and Kaurav asked whether M. tuberculosis could manipulate these fat stores. And remember I said that these fat stores are not just drops of fat, but they have very specific proteins present on the surface, which allow the lipid droplets to execute certain functions, whether it be lipolysis or interaction with other organelles, which would bring about a lipolytic or a lipogenic change in the cell. So asking that question, so before I go into the specifics of the experiment, just like to point out that macrophages have very different lipid droplets from other cell types. So for example, adipocytes are the most well-studied cell types with respect to lipid metabolism, and their lipid droplets can be anywhere between 2 micron to about 70 microns in size. That's a large adipocyte can be 70 microns, and that's practically the entire surface covered by a unilocular lipid droplet. As opposed to that, macrophages have many tiny lipid droplets, 1 to 2 micron in size. And the coat protein that is mostly present in adipocytes is perilipin 1. And in macrophages, perilipin 1 doesn't get expressed, and perilipin 2 and perilipin 3 are the major coat proteins here. So how did they go about asking this question as to what happens to the lipid droplet proteome as a result of infection? So what we did is we took a biochemical approach wherein we purified lipid droplets. And because these are buoyant structures within the cell, when you do a lysis of the cell, you obtain the post-nuclear supernatant by centrifuging that. That post-nuclear supernatant, if you layer at the bottom of a sucrose gradient, as shown here, the end of the other centrifugation run is a layer at the top of this tube, which is a fat cake. So that fat cake contains your intact lipid droplets, and that can be isolated, and you can do many different kinds of experiments with that. So first thing, how do we know that these are lipid droplets? Well, this is a thin layer chromatogram using which we figured out that this was indeed a fraction that was rich in neutral lipids like triglycerides and cholesterol esters. And this fraction was largely devoid of phospholipids, which can't be seen on a TLC, but we can detect that on a mass spectrometer. So we know that the ratio of the neutral lipid versus the phospholipid content is much higher in lipid droplets, and that's what we see in this particular fraction. We also confirm this by using specific antibodies like perilipin 2 is, as I mentioned, a marker of macrophage lipid droplets, and that was clearly enriched in the stock fraction, and the stock fraction was largely devoid of other markers like calnexin, GAPDH, NAMIN, and BDAP2. So what did we find? So if we did a comparison between our study and several other lipid droplet proteomics studies, which ranged from macrophages of another type versus enterocytes, skeletal muscle, hepatocytes, what we find is that there is very little commonality between the proteome of lipid droplets of an immune cell versus other cell types. In fact, our study was able to contribute 99 new proteins to the lipid droplet proteome, although how specific these are to the lipid droplet function remain to be addressed, as do all the other cases of all the other lipid droplet proteins that people have have reported in the literature. So now coming to differences that would arise as a result of infection. So remember, we wanted to find out what are the changes that bacteria is able to bring about actively. So the control that we took in our case was heat-killed MTD, because heat-killed MTD would not be able to actively manipulate something in the host cell, whereas a live bacteria would be able to. So across replicate experiments, we set out to isolate lipid droplets. We then did a TMT-based isobaric tagging approach, wherein we tagged the peptide pools of each of these conditions with a unique identifier tag, and then did mass spectrometry. And what we found is a total of 418 proteins, of which 215 were identified in all the technical replicates. And from this, we chose those that were increased in abundance in the live MTB infected scenario by 1.3-fold or decreased by 0.7-fold or more, with a p-value of less than or equal to 0.05. And we found 86 proteins to be differentially abundant. So now when we tried to use a lot of the online tools to categorize these proteins, we didn't really come up with anything because it's a very small list. It's a very small number to be able to categorize something meaningfully. So we went back to the literature and read up everything that was available on these 86 proteins and then tried to come up with our own classification. So what we found as a result of that is that a large number of proteins do belong to the category of metabolism, in which there's a small subset which belongs to lipid metabolism. So right here in the middle is metabolism. Interestingly, what we found here is that, first of all, a larger number of proteins were belonging to the category of induced upon infection rather than reduced upon infection. So that's why you see more red on this graph. Interestingly, proteins that were only increased in abundance upon live MTB infection belong to two categories. One was protein synthesis. So we found a lot of proteins that belong to ribosomal protein family. And we also found proteins that are involved in vesicular trafficking. Now note that we didn't actually find any lipase in our analysis. So which was a bit of a letdown because we thought that maybe there would be a lipase that would be recruited to the lipid droplets as a result of infection. And that's what's allowing this vectorial transfer of fatty acids to take place. But we didn't. So we think there's more to lipid degradation, and that's what we're working on right now to figure out how these processes could be involved in this vectorial transfer of fatty acids. Just to give you an idea of, remember I said that we took heat-killed bacteria as a control. We also wanted to know whether if we did this comparison between live MTB infected versus uninfected cells, do we see the same proteins or not? So we see about 66 proteins to be differentially abundant in that scenario, about 50% of which overlap with the proteins that we found in the heat-killed to live MTB infected scenario. And two of these also belong to protein synthesis and vesicular trafficking, which we validated by biochemical methods as well. So there's almost equal abundance of RPL11 in the total cell lysate and R-late-B also in the total cell lysate across these two conditions. But in the lipid droplet fraction, we see an enrichment of RPL11 and R-late-B when we have live MTB infecting the cells. So at this point, we really wanted to know whether lipid droplets actually contribute to the growth of bacteria, right? We hadn't even addressed that. So one way of getting rid of lipid droplets to address that question is by depleting triglycerides in the cell. So the question that Nitika set out to ask was, does TAG synthesis in the host promote MTB growth? So for that, she generated a DGAT1 shRNA line in THP1 cells. And what she found is that DGAT1 silencing leads to largely to depletion of the lipid droplets within the host cell. Unfortunately, it did not affect the growth of bacteria at all. So while this was very disappointing, we scratched our heads and asked then what is the purpose of lipid droplets in infection? So we went back to our finding that in vivo, in humans and in guinea pigs, so we had found this in guinea pigs and it was already reported in humans, that solid granulomas, where you don't find necrosis, you also don't find these lipid-rich cells. So the lipid here is identified by Olredo staining. The necrotic granulomas have this cuff of cells, like I showed previously in the cartoon, have this cuff of cells right around the necrotic core of the granuloma. And what was interesting is when we did staining for some inflammatory markers, like TNF in this case, we found that the TNF alpha levels seem to be highest in this region around the cuff, at the cuff of the necrotic granuloma, where the foamy macrophages or the lipid-rich macrophages are also present. As compared to that, the staining in the solid granulomas seems to be more diffuse and lower in abundance. So she asked whether the pro-inflammatory response of macrophages is promoted by triglyceride synthesis. So taking cues from this in vivo finding that there is an association between lipid-rich macrophages, which are called foamy macrophages, and necrosis, she built an in vitro model wherein she treated macrophages with necrotic macrophages in the absence of any bacteria, and the macrophages became neutral lipid rich with a large abundance of lipid droplets, and we call these necrosis associated foamy macrophages. So then she went on to look at the inflammatory response of NAFMs or necrosis associated foamy macrophages to Mycobacterium tuberculosis, and what she found was that there was an increased synthesis and release of pro-inflammatory mediators like TNF alpha, IL-1 beta, IL-6, several others in response to TB infection in the foamy macrophages. So now she silenced DGAT1 in this background, and what she found is that DGAT1 silencing in both the normal macrophages and the necrotic cell supplemented or the NCS treated macrophages Also, in both of these scenarios, DGAT1 was important for triglyceride synthesis. So now she asked whether by silencing DGAT1 and decreasing the triglyceride levels, could she reduce the pro-inflammatory response? And that is what happened. So DGAT1 silencing led to a decrease in TNF-alpha levels in the normal macrophages as well as the foamy macrophages, showing that the triglyceride content of the cell, or maybe the lipid droplets, are contributing to the pro-inflammatory response during infection. So just to summarize what I told you today, MTB infection leads to trafficking of fatty acids away from storage towards bacteria. MTB actively manipulates the lipid droplet proteome, and protein functions affected at the lipid droplet proteome mainly included enzymes involved in lipid metabolism, ribosomal proteins, and proteins involved in vesicular transport. DGAT1 inhibition in macrophages depletes lipid droplets, but it does not affect MTB growth. And macrophage triglyceride synthesis drives pro-inflammatory response to infection. So we're following this up with some in vivo data right now, which we should be putting out sometime soon. And just to bring the findings in the context of what we already knew about Mycobacterium tuberculosis as a master manipulator, what we've been able to add to this picture is the fact that outside of this endolysosomal system, Mycobacterium tuberculosis is able to hijack lipid droplets. And at this point, we don't know what the purpose of this hijacking with respect to specific proteins might be, but that's what we're actively working on in the lab right now. So the questions we're trying to address are, does localization to the lipid droplet affect function of these proteins? Because several of these proteins are proteins that function elsewhere. So ribosomal proteins, what are they doing at the lipid droplet? Proteins like RLA lysosomal protein. So is the lysosomal function of the protein affected? The people who to the work. So the proteomics was done in collaboration and Sneha for I think Devashi.
I think we had some technical issues here. Still waiting. Sheetal, can you hear us?
I don't know when you guys lost me.
No, it's just towards the end of the summary. It was just having that.
I'll just end now. So I'd also like to thank the funding agencies. Can you see my slides still?
No, I think you have to share the screen again.
Okay.
It just got lost after the summary slide.
Oh, okay.
Yes, we can see it.
All right. So, right. So I was thanking all the people who helped us with the work, and also like to thank the funding, particularly the India Alliance, which helped, really allowed us to do this work. Doing this lipid droplet isolation, proteomics, and all that is not trivial, especially in a BSL-3 facility. So I really thank them for all the support for us to be able to do that. I also thank CSIR for their support, which has helped us do a lot of the imaging work. I'm also funded by the Department of Biotechnology for the work that we do on lipid metabolism in mycobacteria. And finally, SERB and Swarna Jayanti Fellowship, which supports me currently for the lipid droplet work to be carried on further. And with that, I would like to say thanks from my joint lab with Vivek Rao's lab, the SG Live lab at IGIB. And I'll take any questions from you guys now. Thank you.
Thank you, Dr. Gandhatra, for that exciting talk. We do had some technical glitches in the beginning, so we deeply apologize for that, but it moved amazing towards the end. As you can imagine, like we got a couple of questions from the chat box, and if you allow me, I can read that out to you.
I can only see one question.
Yeah, no, I can read that out to you. So we got a question from Sweta Bhagwat. Does Mycobacterium use these lipid stores for reinfection or transport to other tissues within the host?
Okay. So to be able to address that question, we need to do this in vivo. So I don't think from the data that we have, it really changes the dissemination of the bacteria. So no, that's not the case. And also when we have an in vitro cellular model system, over time, what happens is initially, even if a small percent, let's say 50% of the cells got infected, over time that percentage of infected cells increases, and we don't find that difference between the control and the D-Get1 silent cells. So I don't think that is getting affected.
Okay. So next question from Shonchari Gorai. Can we mix lipid droplet with lipid soluble antibiotics and target the bacteria to prevent bacterial growth?
Okay, that's very interesting. So there's a group in the UK which has shown that Max Gutierrez group actually, which showed that bedaquiline, which is an ATP synthase inhibitor, a drug against Mycobacterium tuberculosis, actually gets stored in these lipid droplets and is slowly released. So as the lipid is released, the drug is also released in a delayed manner or a prolonged manner. So lipid droplets actually help the drug to work in that scenario. Yeah. But that's very specific to that particular drug.
Right. Along the same line, I think we have a question from Debayon. So lipid droplets uses immune proteins to kill intracellular pathogens and also plays a role in systemic metabolic adaptation to infection.
So what's been shown last year is that in response to LPS, so if you inject mice with LPS, the hepatocytes actually increase their lipid droplet content, and the lipid droplets that are formed actually have some antibacterial effects. So we don't find that the lipid droplets have, in case of TB infection at least, play a pro or antibacterial effect as such. So that may be very specific to E. coli, which was used in that model system, and it may be specific to hepatocytes and not be present in macrophages. So yeah, we don't know which of the cases is.
Okay, no, thank you so much for all this. I'll take one last question before we wrap up this session. This is from Vani. Do you get the lipid profile from the lipid droplet, which has some lipid composition from Mycobacterium? And does this exchange can even happen?
Yeah, we don't know the answer to that question. That is a great question, and we'd like to know that. We just haven't done in-depth lipidomics on the isolated lipid droplets, but that's something we will be doing in the future.
Well, thank you so much, Dr. Sheetal Gandotra, for taking the time to speak in this conference series, and we truly appreciate
Thank you. Thank you.
So with that, we are at the end of Kadambini Ganguly session on health and disease. Please give a big round of applause to our speakers in this session, Dr. Sravanti Rampali Deshpande, Dr. Karishma Kaushik, and Dr. Sheetal Gandotra. It is really great to see all those discussions that are happening in the chat box, and I hope you will keep interacting like this in the next session. So we are having a short break now, and then we will be back at 2 o'clock with the Veronica Rodriguez session on neurobiology. We have Professor Vidita Vaidya and Dr. Vatsala Thirumalai joining us, along with the Jagannathan Vijaya session on biodiversity and animal behavior, joined by Professor Uma Ramakrishnan. Followed by, in the afternoon, we have a special session on gender equity, joined by Shuchita Champak, Nandita Jairaj, Dr. Geeta Chadha, and Dr. Maya Saran. Followed by, at the end, we have the keynote lecture at 7 p.m. Indian time by Professor Gayati Hassan. And I hope you will stay tuned to this and looking forward to other sessions. Thank you very much for joining. Truly appreciate your support. Thank you.
Is it us only?
End the live stream.